Welcome to this explainer. Today, we're diving into something absolutely fascinating, the deep, complex relationship between our DNA and cancer. I want you to think of human DNA as this incredibly intricate biological software program. It's literally the operating system for your body. It tells your cells exactly how to grow, when to divide, and even when to, politely step aside and die. But what happens when that biological software develops a glitch? Well, that's exactly what we're going to explore today, as we decode the highly dynamic world of cancer genetics.
Okay, let's dive into this. For the longest time, humanity viewed cancer almost like this sudden external invader. But modern science, it's revealed a totally different reality. Cancer genetics tells us that cancer isn't really an invader from the outside at all. It's actually the result of a series of microscopic typos happening right inside our own biological code. It's the study of how these genetic changes, whether they're inherited or acquired, accumulate over time, eventually causing totally normal, healthy cells to transform into abnormal ones.
And that brings us to a somewhat chilling but super important question. What actually happens when our biological code makes a typo? I mean, we all experience minor DNA damage every single day. So how do these permanent changes in our DNA sequence, these specific mutations, how do they manage to override billions of years of evolutionary programming and turn a well-behaved cell into a complete rogue agent?
To answer that, let's look at our roadmap for this explainer. We're going to break this all down into 5 parts. 1. The blueprint and the glitch. 2. The rogue agents. 3. Nature versus nurture. 4. Reading the code. And finally, 5. Debugging the system. Starting right off with Section 1, the blueprint and the glitch.
We really need to establish how healthy cell growth suddenly goes off the rails. When normal cells transform because of genetic mutations, the results are, frankly, chaotic. They start exhibiting what scientists call the hallmarks of cancer. These mutant cells basically gain superpowers. They sustain constant growth and completely refuse to undergo normal cell death, which is a process called apoptosis. They just replicate endlessly. And even more wild, they induce angiogenesis. That means they literally trick your body into building brand new blood vessels just to feed the tumor. From there, they invade surrounding tissues, dodge the immune system entirely, and metastasize, spreading to distant organs.
Moving on to Section 2, the rogue agents.
So how exactly do cells get these chaotic abilities? Well, we have to look at the specific genes involved. Think of a cell like a car. Our cells have proto-oncogenes, which normally promote healthy cell growth. That's your gas pedal. But when they mutate into oncogenes, it's like that gas pedal gets completely stuck to the floor. RAS and HER2 are prime examples of this. On the flip side, we've got tumor suppressor genes like TP53 and RB1. They normally prevent excessive growth. They're your brakes. If they mutate, the cell loses its brakes entirely. And finally, we have DNA repair genes like BRCA1 and MLH1. These are the mechanics that fix the car. If the mechanics are out to lunch, mutations just pile up, massively increasing our susceptibility to cancer.
Which brings us to Section 3, nature versus nurture.
Now, what's really interesting here is where these typos in our biological code actually come from. There is a very clear divide between the genetic errors we're born with and the ones we pick up over a lifetime. On the one hand, we have germline mutations. These are inherited from your parents, they're present in egg or sperm cells, and they can be passed down to future generations, which increases the risk for hereditary cancers. But then, on the other hand, we have somatic mutations. These occur after you're born. They're acquired in your body's cells over time. They are not inherited, and you cannot pass them to your kids.
And here is a striking reality that actually surprises a lot of people. Only about 5 to 10% of all cancers are inherited hereditary syndromes, things like Lynch syndrome or hereditary breast and ovarian cancer. That means a massive 90% or more are acquired somatic mutations. To put that into perspective, the vast majority of cancers are linked directly to our lifespan, our environment, and the world we interact with every single day.
So what exactly in our environment triggers these acquired glitches? It's basically a rapid-fire list of everyday exposures. We're talking about tobacco smoke, UV radiation from spending time in the sun, and ionizing radiation. Being exposed to certain harsh chemicals can damage DNA, and so can an unhealthy diet or obesity. Even viral infections play a major role here. Viruses like HPV and hepatitis B can actively disrupt our genetic code and trigger these somatic mutations.
Let's move into Section 4, reading the code.
Because we know what causes these mutations, modern diagnostic science can act kind of like an advanced software developer to spot them. And the technological leap here is just stunning.
We start with basic molecular genetic testing like PCR, which hunts down specific genetic mutations. Then we step up to techniques like FISH to detect larger chromosomal abnormalities. But then we get to next-generation sequencing, or NGS. This allows doctors to rapidly analyze multiple cancer-related genes all at the exact same time. And the absolute cutting edge right now? Liquid biopsies. We can actually detect and monitor circulating tumor DNA simply by drawing a standard blood sample. It's pretty mind-blowing.
And finally, Section 5, debugging the system.
Spotting the typos is one thing, right? But fixing them is another entirely. How do doctors actively fight back? Well, this brilliantly illustrates the true power of cancer genetics, targeted therapy. Instead of just carpet bombing the body with traditional treatments, personalized medicine lets us exploit the tumor's specific genetic weakness. We can design drugs that exclusively attack the genetic abnormalities in the cancer cells while totally sparing normal, healthy cells. A perfect example of this is targeting the mutated HER2 gene in certain types of breast cancer, effectively turning off that stuck gas pedal we talked about earlier. And honestly, this precision oncology, it's just the tip of the iceberg. The future of treatment is bursting with optimism. Cutting-edge tools are opening the door to completely individualized cancer care. We're looking at experimental gene therapy and CRISPR-based research to physically go in and edit or repair defective genes. We're using AI-assisted diagnosis to catch incredibly subtle patterns that humans just can't see. We're even developing individualized cancer vaccines based on a patient's unique genomic profile. It's a real game changer.
So, what does this all mean for you? Well, for anyone with a strong family history, multiple affected family members, or early onset cancers, genetic counseling is an absolutely crucial resource. Counselors can help you interpret these advanced tests and really understand your risk. Knowing your own hereditary blueprint gives you the immense power to take preventative action. It means utilizing early screening to catch any potential glitches long before they can cause a system crash.
We've seen how a few microscopic typos can turn our biological software against us. But we've also seen just how incredibly fast our diagnostic and treatment tools are evolving to fight back. So I want to leave you with this thought. As AI-guided therapies and CRISPR gene editing rapidly rewrite the rules of modern medicine, are we finally on the verge of completely outsmarting cancer's genetic code? The blueprint is in our hands. Thank you so much for joining me on this explainer, and I really hope this sparks your curiosity to explore the amazing world of genetics even further.